All right, we're getting the last few colleagues. I hope you got caffeinated and are excited for our panel here. Okay, so welcome to this panel, the full-on panel in this morning session of discussing new trends in science and medicine. And we have had on many of the Galien forum dialogues around new modalities that allow the shape of medicines that break out of therapeutic limitations and boundaries. And we have discussed quite often either genetic medicines or how you can treat diseases where there may be genetic drivers. While previous years, it's been a lot on focus on what's the next technological step, a sophisticated technological step to overcome barriers. We would like to widen the perspective now. How do we become somewhat more patient-centric in using the insight of diseases where there is maybe a single augmented or reduced gene causing troubles for patients, and adjust rather than make more technological and sophisticated our current best modality. And you will hear how we can, on one hand, be very pragmatic in deliver genetic medicines or medicines to treat diseases with a genetic link locally, how we can think about further improving to get more of the medicines to the right site of action, and how we then move from learning from the more rare traditional genetic diseases to more treat common conditions where there may be still a single driver. On the positive note, I think the regulatory environment has actually moved from somewhat more cautionary stand to realizing that it's time to advance in a rigorous manner and be innovative also in the regulatory and the national scientific platforms of US, how to move this to transform patient lives. So I've been part of initiative cross industry, NIH and FDA, what we call bespoke medicines, where we try to target patients that have a very specific need and look at ways of sharing platforms. And we heard about, I think, UPenn's ability to individualize medicines for saving a life of a baby. There is also now at FDA, on a very positive note, an aspiration to accelerate things that are strategically important for the medical community. And I think a part of that is to really facilitate single-arm trials in rare disease, if they come with an ambitious way to measure and follow up patient registers, because it's difficult for some rare disease to do studies and have placebo controls. In other areas, I've noticed there's regulatory excitement to look at chronic illnesses, and if you have approaches that are deemed safe within current knowledge, to look at ways of chronic illnesses to have biomarkers that could allow you to register a new treatment faster, particularly if they come with modalities that can include local delivery to allow us to address some of this dramatic burden of disease. And then, of course, underpinning on this is how we can scale availability of medicines, whether it's to underserved community in our country or to patients worldwide, which means thinking about moving out of very fragile treatment paradigms that need complex cold chains to things that can be delivered in a more broad sense. We have a very, I think, distinguished panel here. Next to me, we have Judy Lieberman, who is the big brain here. Not to underestimate either myself or my colleagues, but you represent the academic thinking on how we unleash the full power of some of these new modalities. And I know you have been part of the early efforts in RNAi, and you continue to push innovation, how RNAi can go from the liver to more tissues. And I think you have been interested in brain delivery, which is one of the new really important frontiers, and where we have seen some transporter proteins be very revealing in what you should think about the future. Cedric, Francois has a career as an entrepreneur. I think you started actually in surgery and then moved to complement diseases, and really represents how pragmatic solutions in understanding complement disease can deliver patient-available treatments now, and how you can target for different diseases local or systemic available medicines in the C3 complement. We then have Avista Therapeutics here that are focusing on diseases of the eye, where we had one of the first approvals with Spark and Kathy High that I have had here many times as a guest. And I think you are taking that experience to a new level, and what's very exciting, you benefit from local delivery, but I think you are trying to understand the different cell types, including in the subretinal space, that we need to curate or heal, and how you can improve delivery to make it most efficient for the root cause, not only at the gene, but the cell. Devyn Smith, you are CEO of Arbor Biotechnologies and work with innovations coming from the MIT, Harvard laboratories of Feng Sang that relates to novel, more compact Cas CRISPR technologies you can put in a larger payload. And you took a bold decision recently, moving from the more low-hanging fruit of liver disease, where you partnered, and are, I think, taking on the more transformational opportunities with your company over the next few years. So we look forward to listen to what made you go from the low-hanging fruit to the more exciting but higher up to climb fruits at the tree of human disease botanic gardens. And then at the end we have another example, what I find, of a bold CEO and entrepreneur from Pacira BioSciences, Frank Lee. And similar, you have established a company that contributes many recombined, reformulated pain medications for patient benefit. And who could think, once you found such a good business rhythm, that you would return to entrepreneurship and start to think about solution for osteoarthritis by local gene delivery. Again, I think taking a foundation in your business and becoming bold. So we're up to, I think, a real treat here. So Judy, tell us with a few words what you think today's technologies can deliver at best, and what you like to see when it comes to pragmatism, and where is the next innovation going to take a leap for us in diseases that have some genetic burden?
Okay, so my expertise is really in RNAi based therapeutics, and and I think there's still a lot of room to make it, to apply it to many different diseases. So there are now, so in 2001 was the first demonstration that RNAi operated in mammalian cells. And I got involved at the very beginning and showed for the first time that you could actually use small RNAs as a drug to protect mice from death from autoimmune hepatitis. And then I was involved with Alnylam, which has made 5 of the 6 drugs. It's a real success story in RNA therapeutics, and I think it became a success story. I mean, there were hundreds of companies formed around the same time as Alnylam because they really took seriously what were the obstacles to making it a drug. And delivery is really one of the big issues for all the genetic therapies, including RNA therapy. So in terms so initially Alnylam and a lot of the other companies focused on rare genetic diseases, and they focused on the liver because they had an amazing chemist who figured out how to very efficiently deliver small RNAs into liver cells using the asialoglycoprotein receptor. But they haven't really moved too far beyond that. In recent times, there's an academic, Anastasia Karova, and Alnylam have both developed very efficient ways of delivering small RNAs into the brain and eye. So I think that is, and it involves just cholesterol conjugating RNAs, and they've been able to show that if you start with the mouse that has a really small brain and you want to go to humans, it's a really much more challenging, and I think they've solved that problem. So I think that's one way to go. I think Alnylam has also the liver is the manufacturing factory of the body, so it's not a bad target, but a lot of diseases don't involve the liver. I believe now there are technologies that are very effective at introducing RNAs in a very specific cell type manner into different cells. And we developed the first targeted delivery, which involved an antibody fusion protein that bound siRNAs and showed incredible selectivity. We could knock down genes in all lymphocytes, and lymphocytes are the hardest cell in the body to target, or only in activated lymphocytes, or only in certain subsets like T regulatory cells. So I think antibody-mediated delivery is one way to go. It's recently Avidity, which develops antibody oligonucleotide drug conjugates, was bought by Novartis for a huge sum of money. So at least there's some faith that is the way to go, and we have so many antibody drugs and more antibody drug conjugates, so I think that's very promising. I've been developing a way that uses an aptamer, which is like a nucleic acid antibody-like compound, and with an aptamer to the first tumor antigen, EpCAM, which is only 19 nucleotides long, you can make an RNA so you don't have any of the problems of lipid nanoparticles, which are very toxic, and can be chemically synthesized, that has similar specificity. And we've mostly been focusing on cancer. So we have this EpCAM aptamer. EpCAM is expressed on all the epithelial cancers in their stem cells. And we're getting amazing immunotherapy results with that technology.
It's so fascinating to listen to you on how we basically create hybrid modalities, antibodies that are proven, link them to RNAi, and some link them to the nanoparticles and can also infect immune cells. And you mentioned to us, you know, a lot of us heard about ADCs, antibody drug conjugate. Now it's AOC, antibody oligonucleotide conjugate. I hope you forgive me for a joke here. I do hope AOC. That this AOC will be much better for New York than the other AOC. As you know, we lost a lot of high-tech jobs in New York because of the other, but Judy has promised me to start some AOC companies here so we get the jobs back. It was just a joke here, to warm you up, an icebreaker. Next time I'll joke about the right wing of this country. All right, Cedric, you have taken on a target that seems to play a critical role in many complement-mediated disease, C3. And I learned from you that the job of neutralizing C3 comes with some intrinsic challenges. And I think you are approaching it in a very elegant way, both to incrementally take it forward and to think big about how to keep improving patient benefit. Could you share with us what has driven your entrepreneurship in this space, which has transformed opportunities for patients with complement disease of the eye or the kidneys and remic syndromes, and gives hope for the future as we discover that complement are actually involved in many more diseases than we originally knew in the textbook of medicine.
No, thank you, Mikael, and it's so wonderful to be here. Yeah, I think with complement specifically, and in the context of the subject of this panel, what was really fascinating for those of you not familiar with complement, it's the oldest part of our immune system. It's about 30 serine proteases interacting with each other, actually quite similar to the clotting cascade. And it is our oldest form of immune surveillance. So 400 million years ago, when we were jawed fish in the Ordovician period, we didn't have T or B cells yet. And what organisms did at the time is they would have a lot of C3 in circulation that would put out a little tail and bind covalently to all the cells in the body. So it was like a paint job, if you want. And the purpose behind this was that a cell's purpose was to immediately inactivate that paint, internalize it, and process it. Now, cells that did not clean up after themselves got ruthlessly eliminated by macrophages, and that was the immune surveillance. So if you had a cell that couldn't clean up after itself because it was sick, infected, whatever it is, macrophages, who are otherwise blind, would see that product on these cell surfaces and remove it. Then obviously afterwards we got T and B cells, we became much more sophisticated with self versus non-self. But this old immune surveillance system that sits in the background is still there, has a lot of crosstalk with adaptive immunity. And especially when we grow older, starts playing an important role in many pathologies. And you know, when I explain this to my father or my siblings, I always say, like, you know, I like to think that when we grow older, much older now than we used to, we get less good at separating water from lipids. So lipids kind of slug around. They don't sit in the fridge, so they oxidize. And oxidized phospholipids in the interstitial spaces create kind of a broad-based, up-leveled state of immunity in which complement plays an important role. And what that means is when you get more complement deposition, cells have a harder time cleaning it up, and that is associated then with several of the pathologies that we are pursuing. But where it became quite complex is when you think about it in the context that I just said, is how much C3 is in the body. For the scientists in the room, 7 micromolars of a 187 kilodalton protein. That's a proverbial F-ton of protein. If you want to control that, if you want to remove it with antibodies, You'll appreciate this, Judy. I mean, that's
lot.
virtually an impossible task to accomplish. So what we did is we said, what if we approach this from the angle of putting this enzymatic cascade on ice? In other words, take a enzymatic approach to an enzymatic system. So we don't try to remove all C3 from the body. Instead, what we do is control it from getting out of control. And it's a very neat and interesting way of approaching this cascade, which we have now applied with an approved product, the first approved product for geographic atrophy, which has been on the market for 2 years now, and then Empaveli, which is a drug that's approved for 3 rare disease indications.
That's terrific. I guess I have to say a joke on the other side, not to be blamed here. As we are in November, we prepare, our wishes for 2026, and having been part of the community of scientists that developed the COVID vaccine, I wish the ability to do math is improving at certain parts in this country when you end up the tens of millions saved lives in the globe by these vaccines versus the few that in some way had moderate to minimal harm. I just hope the math will turn us back into where do we have benefit versus risk in the outcome. Math is good because it's exact science. Politics is not as exact as math. So cheers for America becoming a math power in the future. So you see, I'm absolutely neutral. I'm just trying a few icebreakers with you guys. Now we turn to Avista Therapeutics, and I'm intrigued to listen how you're really trying to mix delivery, rare genetic disease, with still trying to improve where these genes act in the eye to make sure you avoid the risk for inflammatory and other reactions or not getting the gene expressed in the proper space. You're really being a high-tech ophthalmologist here. Share with us your story.
Sure. I'm Rob Lynn, the CEO of Avista Therapeutics. And at Avista, we were focused on the improvement of delivery of genetic medicines. And you may say, well, delivery seems relatively mundane compared to some of the really exciting work that's being done on the RNA front, or Cedric and his complement cascade, or even some of the gene editing that Devyn will tell you about later on. But I will say to you, if your product does not get delivered to the right place, it will cause some problems. And I don't know if anyone else has had this issue, but if you order a brand new exciting toy for the kids and Amazon doesn't deliver it to our house and it goes somewhere else, that causes big problems because the product doesn't get to the right place. And I don't know if I'm the only one who does this, but sometimes I'll take a look at the picture where it gets delivered on my phone and walk down the street trying to figure out whose door that is that it actually got delivered to, and try and find it. But I think that therein lies the challenge of delivery. And I think one of the delivery workhorses for genetic medicines has been AAV, the adeno-associated vector. And it works mostly, not terribly specifically. And as Mikael was saying, the first gene therapy delivered in the eye was delivered subretinally with a AAV vector that was a stock AAV that nature has created. What we wanted to do at Avista was take what nature has created in the AAV and improve it by engineering the capsid to try and engineer additional properties into it. And really what we wanted to do was make something that's more specific to some of the cells in the retina and improve delivery by lowering the dose, because we know that when you deliver things intravitreally into the eye, that there's a dose-dependent immune response. And so by lowering the dose, we thought that we could lower that immune response and improve efficacy, and also somewhere along the way, the accountants like it when you can reduce the cost of goods as well. And so the goal there was to create better vectors that allow you to reduce that dose. And I think our latest version, which we have in a partnership with Roche right now, has over 3 logs efficacy better than the stock AV2 when injected intravitreally. So again, enabling more precise targeting of those medicines by lowering the dose and hopefully reducing some of those reactions of the immunity that we're seeing that cause some real tragedies, especially in the systemic dosing when you're not dosing locally. One thing that came out of this is that we became an accidental AI platform. So I think we heard this morning at the first panel that with the models and the GPUs that NVIDIA has, and would really like to be in that business instead, but things being the way they are, we have the GPUs, and we had hundreds of thousands of data points from our screening of AAVs. There are a dozen that work really well, and then a couple 100,000 that go from not working to working a little bit, and then trying to figure out what happens there. And so a lot of it is just pattern recognition. If I came to each of you and said, hey, there are 2 things on this table that taste really good and 10 that don't, you might be able to say, hey, I can find some commonalities between them. But when we're looking at hundreds of thousands of lines of data, that's where the AI models come in, and the machine learning really is able to pick up what's good there. And so really thinking about how we can take things beyond the eye and look at dosing in other tissues, such as the brain or systemic dosing, really taking some of the lessons that we've learned through the hundreds of thousands of high-quality data lines that we have, and using that to hopefully create better AVs for other applications as well.
Thank you, Rob. So, Devyn, we heard among other things Rob's advances with Avista to lower the dose and more specific targeting. And he spoke about the AAV workhorse. More recently, we have had a lot of optimism about moving from viruses to LNPs. You shared with me that very recently one of the LNP companies had reported for a very promising treatment a severe case of side effect, just underscoring to us that the dose probably matters, and these are still days of being prepared for unanticipated effects. So you work with both of these systems, and I know recently you are considering to focus on new organs beyond the liver. Tell me a little bit about how you think about these choices and your boldness to move into the more rewarding but harder to get to success areas.
Yeah, thank you, Michael. So at Arbor, we are focused on the cargo exclusively. The company was co-founded on an idea with no IP seeded from Fong's lab or David Walt, the other co-founder, and that was to go out and identify novel differentiated gene editing approaches, and really approaching gene editing as you approach a modality, which is you have a library or a toolbox of approaches you can screen against a particular target of choice to find the best approach. That's kind of the underlying ethos. So we have a range of editing material that is all small, so that we can easily, for example, fit an RT editor into an AAV, or rewrite larger sections of DNA, within an AAV, for example. So we become delivery agnostic, and we purposely have chosen not to build a delivery platform. And the reason for that is there have been billions of dollars put into companies like Rob's and others to do exactly that. And by actually leveraging all of that, we can actually identify the best approach, best delivery modality for our particular indication. We started in the liver, as Michael said, using LNPs, which are safer than AAV for liver-targeted approaches. Not perfect, but they are safer than AAV if you're targeting the liver. And that's where all the gene editing companies are today, for the most part. And as we looked at this space, you have 2 options. You can go after the same targets as everyone else is going after, which are lower risk, or you can do something different. So we decided to go do something different. Because to really recognize the true value of gene editing, we've got to move outside the liver because there are hundreds of other diseases we can target. So we have partnered with Chiesi Pharmaceuticals, our lead program, which is in the clinic in the liver, in a rare pediatric disorder, and really are turning our attention to other areas. And one area in particular is the CNS. Now, the CNS is a graveyard of failed molecules and compounds over the years. However, one of the distinct advantages of gene editing is that you really can go to the true root of whatever target or pathway you want to hit. That is an advantage that we've never had before within the CNS. In addition, over the last decade, the biology for many CNS diseases has become much more identified thanks to efforts of all the various academics and folks like Judy. So we have decided to, we are focused in on ALS initially. There's been tremendous efforts to understand the biology there. There's opportunities for gene editing to really alter the biology in that disease. And to your point on delivery, there are multiple delivery approaches available from an AAV perspective. One can do local delivery, one can also do a systemic capsid. And so for us, delivery is critical, but there are so many approaches that we can really identify a best approach that fits for whatever target we're doing.
Given the recent success reported by Unicure for Huntington disease, where they injected directly into the cistern of the brain, I think, right? Doesn't that make you intrigued to look at it as a good first approach to get CNS accumulation?
Yeah, I think particularly for ALS, where we select ALS for that reason, because the spinal motor neurons are imminently accessible when you can access them via some sort of intrathecal approach. The ASOs that are pursuing ALS are using monthly intrathecals because you can access the spinal motor neurons. So we like that local approach because it is manageable. It's a region that you can actually access. Unicure's data is very exciting because the ability to go beyond the spinal motor neurons into other regions of the brain, and that's something we certainly are looking at closely for additional indications. But I think it does suggest, despite all the headlines around AAV, there are approaches that allow you to do so and do so safely today.
And then Frank, I mentioned in introduction that I was really excited to see how your business strategy that has been successful in refining and combine existing medicines for pain and mainly small molecules that were reformulated and recombined. And from that good hard work, you as a leader are looking at one of our really major health burden, osteoarthritis of the knee, leading to a lot of surgery, weeks of recovery sometimes, and you're approaching it with gene therapy. So tell us what made you move from a comfortable zone into that space, and whether you needed to have some pretty tough fights with your leadership team or the board or the finance community, or they all just embraced your courage here?
Well, thank you for that, Michael. I have to say I was inspired by Father Judy this morning when he challenged us to say, hey, how do we benefit the broader patient population as opposed to the smaller ones? And so, to be honest with you, when I first took this job and we were looking at the portfolio and I ran across this gene therapy, I said, absolutely not. We're not doing gene therapy going forward. But as I looked more closely at it, I became intrigued by this possibility of gene therapy for the masses as opposed to gene therapy for the few. Okay? So let's talk a little bit about the science and more importantly about the patient journey and what needs to happen. So the science, it's a high-capacity adenovirus platform. What it does is it helps the body produce IL-1RA. So as many of you know, that IL-1 is an important inflammation cascade in the body. There's already a drug approved called anakinra that blocks IL-1. It turns out, though, that the half-life is very short. It's only about 4 hours. And so we thought about that, the team thought about that, and say, hey, how can we improve on that? And so what this does now is using this, let's say, high-capacity adenovirus platform, which can hold 30,000 base pairs, you inject it locally into the knee, and over time, the cells in the knee will produce IL-1RA when it needs it. So there's an inducible promoter. Okay, so the phase 1 studies have been very encouraging. We're in phase 2. But now let's step back a little bit. So what are the pragmatic challenges and opportunities of gene therapy for the masses? Well, it's got to work within the system. So if you think about osteoarthritis, and many of you may know people with osteoarthritis, your family members. You may have it yourself, so you'll know. So typically, you'll be on this journey. You'll be on this journey for decades. You'll go from NSAIDs to various over-the-counters. You'll pay $2,000 for insoles for your shoes. You'll do anything to avoid surgery. And so along that time, you'll go through many, many different kinds of medications, and unfortunately, of late, there haven't been much in the way of new medications to help osteoarthritis patients. And so when you do get something, it's an intra-articular injection. So there are plenty of physician offices that provide that, know how to deliver that. So the system is there. They know how to bill for this and get reimbursed for this. So again, the system is there. And as we've looked at the marketplace, the current treatments provide about 3 to 6 months of benefit. So we know from talking to patients, payers, physicians, that getting patients out to a year is considered transformational. And beyond that, wonderful. But so that's what we're thinking about, is how can we get patients out that far, at least that far? As we think about that now, we have to think about, from a patient perspective, what that feels like. So from a gene therapy, I'd love it so that gene therapy becomes just therapy going forward. So there's not this sort of experimental halo over gene therapy. So they know that by injecting very small amounts, it's very, very safe, number one. Number two, you need very small amounts of it, so the cost of goods, it's very attractive. And that allows us now to go to payers and others in the healthcare system and say, hey, listen, this is more like a traditional drug as opposed to hundreds of thousands of dollars, millions of dollars. So that's what we're striving for. And of course, as we think about that journey, for chronic diseases, we need for now this therapy to be redosable over time. And as folks also have osteoarthritis and other joints, we need to think about what that does now. So do the level of neutralizing antibodies make a difference in terms of how these kinds of treatments play out in the body? And so those are important questions that we're going to answer in later stage studies. But we're excited about the promise of being able to deliver gene therapy to the masses. And I know there's been a lot of discussion about gene therapy of late, but I have to tell you, I'm very excited about what's ahead.
Thank you so much. We'll take an opportunity to open up for questions, and then we'll come back to the panel if there are any questions, as you've got to know our 5 knowledgeable, accomplished guests here. Yeah, do we have a microphone here?
It's a little bit broad than what you are focusing on for gene therapy. So I'm a very fierce patient advocate for my very own genetic blood disorder called Cooley's anemia, also known as thalassemia. And we have Zynteglo, so happy, Bluebird Bio, which is now rebranded back to the original name. I forget the original name. And Casgevy, Vertex. That's fantastic for us American patients. And Zynteglo is at $2.8 million. So they realized they cannot get that in Europe, and they shut down their European operations, which was very hurtful to the patients in Europe. I'm thinking, how do we get this to Bangladesh and Pakistan and Iraq? These are the patients that are devastated, not me. I have my 1 to 2 pints of blood every 14 days, safe, no HIV, no Hep C. In India, they have blood, but they don't even test for HIV. So last week, we had 12 patients that were affected with HIV in their blood. So how do we get gene therapy to these countries? Affordability, accessibility. I know it's a little bit out of your realm, but just wondering, because you guys are still in the biotechnology space.
I'll say a few words, and then I welcome anyone. I think it's a great question. Clearly, some of the interventions we heard from the panel, like injection into the knee of a viral vector encoding IL-1 receptor antagonist, or to defective genes into the eye, or Synfovre, the blocking of C3 in geographic atrophy disease, has the potential, because it's a relatively less intrusive procedure. I think what's the challenge with some of these treatments where you need to harvest blood, take it ex vivo, transfect it. So it is a pretty demanding question. Is it possible to bring it to the world? Or do we really need to turn the innovation to the next level? And we heard a little bit about that. Make it a simple injection where the mRNA targets the right precursors to the erythrocyte and are curated in vivo. I'm not sure whether these are enough, good enough for large application modalities, or whether they are an important stepping stone. But I let others pitch in here.
Sure. I spent a number of years at the Bill and Melinda Gates Foundation working on these issues, and they're not easy, and thank you for bringing them up. As the father said earlier this morning, that part of our mission is not just to create life-changing therapies, but also to make sure that they're distributed. I think he mentioned, making sure that everybody was able to access them. And so I think, as Michael said, part of it is new technologies. Part of it is trying to figure out different ways of manufacturing. It's really interesting. So when I joined the Gates Foundation, I went from making cancer therapeutics for $100,000 a year to trying to figure out how to get a vaccine for meningococcal disease under 50 cents, because that was the target product profile, and we came in at 42 cents and still heard from way too many people that was too expensive. But trying to figure out how do you do this. Part of it is, as we said, the new technologies. Can you find better therapies that you can create for a lower cost of goods that you can pass along, and then other ways to really think about stripping down the manufacturing process from the ground up to make things at scale, but also be able to customize those to the right diseases. And so I think there's hope, but there's big challenges.
Jeffrey Lieberman at Columbia University. I'm a psychiatrist, and blood-brain barrier penetration has obviously been an issue. You described using RNA approaches. Probably a more blunt instrument approach was developed by Elizabeth Hillman using focused ultrasound. Other approaches that have been used to try to methylate or deuterate or modify the biochemical structure of from the drug to do this. How do you see this fitting together? I mean, is it going to be a modality that you're working on that could supersede all the other efforts to do so? And also the other thing is, what is the duration, the durability of the effect of the approaches that you're taking?
Yeah, so one of the biggest...
By the way, no relation that I know of, but...
No. So one of the really big advantages of siRNA drugs is that they are incredibly durable, like treatment for amyloidosis or cholesterolemia. In non-dividing cells like neurons or microglia, some RNA drugs now are given every 6 months. So I think, RNAi is here, so we have drugs, so it's a good place to start. I think gene therapy will eventually, genetic engineering will eventually become what, more feasible, less scary, but in the meantime, I think the durability really is very impressive. And that durability only came about because people really, really studied the chemistry of these molecules and figured out ways to increase the activity like 50 to 100-fold. And same thing for toxicity, like the first version of the drug that's used to treat amyloidosis, the trials were stopped because people died, and getting the dose down, 100-fold is really important. So I think it's a matter of really staying focused on what you have to accomplish to get the drug to do what you want. I think the brain is probably, maybe it's the most challenging place to go because I don't think we completely understand the blood-brain barrier, how to get around it, although intrathecal type injections, I think the technology is there now that I want to try RNAi for neurodegenerative diseases because we showed that there were these molecules that cause inflammation in both neurons and microglia that when we inhibit them, we can really protect mice from serious neurodegeneration.
So Judy, there's been these discussions about the brain shuttles, like transferrin, I think IGF-1. You feel they are ready to be exploited to transport oligonucleotides to the brain?
I don't know. I'm not enough of an expert. Okay. I think people have come forth with ideas that haven't proven to work.
Rob?
Both for AAVs and also large molecules, people have used the transferrin receptor to cross the BBB. There are AAVs that are engineered to target transferrin receptor. It really depends on the indication that you're looking for. If you're looking for broad transduction or entry across all brain regions, I think that is something that is useful in that. If you want to target a specific brain region, I think that's where the focus ultrasound comes in, and we've been doing some experiments with that where you're able to use the focus ultrasound and open up almost a Stargate portal through the BBB that lasts anywhere from 4 to 6 hours, and then you have crossover to a targeted brain region, and I think that is, again, indication specific where you want to get your targeted. We have one more question here.
Thank you. This is related to the previous question about accessibility in global markets. What are the regulatory ramifications? I apologize. I'm Omar Sarang with DNA Nexus. We've worked with the FDA for the last decade to enable efficiencies in regulatory review. And when I think about gene therapy and cell therapies, it just seems profoundly challenging to get through the regulators. I'm just curious how this stuff gets reviewed. I mean, you go through your clinical trial process, but I really am, I don't know, I'm a little overwhelmed by how regulators will be able to adapt this, and not just the FDA, but global regulatory agencies.
Well, I can speak to it at least from a gene editing perspective where things stand today. A couple of years ago, the first gene editing INDs took well over a year to clear because the FDA was still trying to figure out what do you do with this, because you can't stop therapy once you've dosed the patient, right? It's done, and you live with whatever consequences there are. Flash forward now, I think the agency has become a lot more comfortable after seeing many of these. We filed an IND late last year and cleared in 26 days, so first company to do that for gene editing, and we did not go ex-US. So other companies have gone ex-US first, brought clinical data in. So the FDA, I think, is much more comfortable. I think they're also recognize, like baby KJ, for example, earlier this year, that was done in record time with minimal traditional CMC and quality approaches that one would take with a traditional. So I think the FDA itself is becoming much more flexible to think about how do we get access to these potentially life-saving therapies to more people in a safe but also a much quicker process. So I think that's good. And ironically, you're starting to see that spread. The Europeans are looking at this heavily, as are the UK. And so I think that is beginning to help broadly this sort of one and done world of we've got to figure out how to give people access, because these therapies, once they're there, patients don't want to wait 7 years for this thing to get to the. And so I think you're seeing a real, revolution within that space, and it's one to watch very closely, because you're walking a very tight line between safety and efficacy here. Every time you get a safety event, then everyone has to take a pause. But
and I think your optimism is really warranted. I also need to acknowledge that the current FDA administration seem to be thinking in a more flexible manner than we have been used to. They have released strategic vouchers to make new areas be reviewed in 2 months. And they are looking at unusual ways for difficult areas to get approval, including biomarkers. So I think it opens up for a positive change, including we have seen, which sometimes is good and sometimes bad, that FDA traditionally, it was the reviewers that decided the outcome, and the commissioners would basically only intervene if it looked like someone was, hallucinating. Otherwise, whatever outcome of a reviewer would stand there. I'm actually quoting one of the former FDA commissioners. I won't give his name. We have seen this administration, which I think is intriguing, overrule reviewers, and sometimes we may like it and sometimes not. But at least it moves the outcome from maybe 1 or 2 individuals far down in an organization to opportunities for intervention. Of course, it's nice if we can have a process for intervention, but I think it's a way to change a very traditional system. Just to close this session, I thought we walk through this panel and just ask you, if we all were invited October 30, 2030, I don't know, it depends on how well you perform now, if we get invited. What would you think we will be talking about in this field as a recent accomplishment? And what, if anything, would you have liked yourself to be involved in? Since we have 2 public companies, I will say a forward-looking statement that any statement here is linked to uncertainties and doesn't necessarily reflect future outcomes. This is an exploration and not an aspiration by any of the panelists. So I hope that will protect you all. God bless you. Start with you here.
I don't know, that's a difficult question, but in my field, I would like to see targeted RNA being delivered throughout the body and being used to treat diseases that are untreatable, because basically you can go after any gene. You can do it pretty quickly if the delivery system is in place.
Yeah, from my perspective, obviously AI. I think that, again, I was sitting with my team a couple of weeks ago, and I said, how do we file an NDA in 2 weeks instead of 5 months, right? And I think that should be possible with the technologies that we have. I think that the way we run clinical trials should, by any account, especially with once and done treatments, right? We should be able to do that in a real world setting. I mean, we should be able to track cognitive decline, motricity, vision, etc., through the tools that you use right now, and be able to have large population cohorts that you send out, and then with a Bayesian approach very quickly get answers as to what is going on. I think that developing a drug should not take 7 years, and I think we have all the tools available to do things much faster while still maintaining a good view on safety, and do it much cheaper. And then to the point of global access and medicines becoming cheaper, for medicines to be cheaper, they need to be cheaper to make, and they need to be cheaper to develop. And I think that's the window that we have in front of us.
I'm super excited again to see the innovations in the clinical trial part of our business. I think we've gotten so many innovations in the preclinical part with the AI work and looking at design there on different molecules and how they might perform in silico. And I think taking some of those advancements in the clinical trial stage would be just amazingly helpful, because if we think 5 years from now, if we're on the current trajectory, we're just going to be able to see where the innovations of today or a couple years ago are going to get us in human trials. And the field's advanced so much in the last couple of years that I think we'll have super advances to test in humans over the next 5 years.
I'd love to see gene editing move beyond treating patients once they're symptomatic to actually preventive. So for example, a patient who has Huntington should not have to wait till they exhibit symptoms to be treated. If you have the ability to actually remove that mutation 20 or 30 years earlier. Now, how you do a clinical trial on that, how you get that paid for, all those pieces, very, very challenging. But if we have the technology to do that, we have to find a way to do that. So.
Well, as we enter the next decade, I'd love to hear gene therapy referred to as therapy. And what that would mean is that it would be considered safe, viable, accessible, and truly delivering value to such a wide, large number of patients worldwide.
Well, thank you so much. I want to first thank the audience. You seem very attentive, engaged. I could see no one falling asleep. I thank you for participating actively. Thank you to this panel. I think they represent 5 amazing entrepreneurs. You could hear their vision, their pragmatism. So I think we will, by 2030, feel very proud of what we have accomplished. And maybe, as you were asking, we could have global launches all over the world for most of the treatment. So thank you, everyone.